Fusion, the international science radio show.
I'm the professor of RNA biology at UNSW Sydney.
Yep.
I was previously Chief Executive of Genomics England, and then prior to that, Director of the Garvan Institute of Medical Research, and that's probably enough.
Okay. What's the misunderstanding in molecular biology?
Well, as most of your audience will know, the paradigm that emerged out of the double helix and the studies in the 40s, 50s, and 60s, mainly studying bacteria, was that genes make proteins and through the intermediate of RNA. So there's a gene at the DNA level that's transcribed to produce a temporary copy called a messenger RNA that's then translated into protein. And there's a gene for hemoglobin to carry oxygen, there's a gene for insulin, and so forth. So the understanding that grew out of those early studies using on microorganisms, because we were too difficult to study, was that genes make proteins and we are composed of the proteins. They're the basic components or Lego parts of cells and organisms. And that's largely true for bacteria. They're relatively simple cells and most of the genes encode proteins. In the 1970s, probably the biggest shock in the history of molecular biology was discovered that most of the human genome does not encode proteins. Only about, with regulatory elements, only about 2% of the entire DNA specifies proteins. And people thought, what's going on here? Now, there's a longer intellectual history, but basically they jumped to the conclusion the rest of it must be junk. It was sort of evolutionary debris. It was informed partly by the fact that about half of the genome was composed of repetitive sequences. In other words, sequences repeated, like saying automobile 1,000 times, which they just assumed was junk. And there was a cogent intellectual history to this, but it was also just an assumption because it was outside the conceptual framework. Everybody assumed that all genetic information was transacted by proteins, and that's all she wrote. Now, the misunderstanding at that level was to assume that the mechanisms that regulate microbial physiology will be the same as those that regulate human development, or that the way that human development is regulated and controlled will be limited to those mechanisms that control bacterial physiology. So this was believed for a long time that most of our DNA was junk, just evolutionary hangover. However, the second shock was then to find out that most of this DNA, which is not made into proteins, is actually made into RNA. So it's RNAs, but the RNAs are not translated into protein. So for a long time, people said, oh, this is just transcriptional noise, it's background, because most of these RNAs that are produced from the rest of the genome are very cell-specific. They're lowly expressed. They seem to evolve very quickly, so they're not highly conserved, which is the gold standard for function. And so there's been a controversy for the last few decades. I think it's resolving now, that whether this was just noisy junk or whether there was something else was going on. Where I came into this in the late 1970s was when they first discovered most of the genome was being transcribed. And I thought to myself, well, there's two possibilities here. One is that it's junk. The other is that there's another form of information that we hadn't expected. And so that drew me on because that was interesting. And in fact, it was more plausible if you thought about it. So anyway, what's transpired is that most of the genome is actually specifying genes that produce these what's called non-coding RNAs. They don't code for proteins. And these RNAs are responsible for organizing our genome during development. Now, if you stand back from it, Ian, from the moment of our conception to adult, you have to have trillions of cell fate decisions. An adult human means various estimates, but around 30 to 40 trillion cells. And, if you start with one, divide into two, and then to 4, 8, etc. Imagine that That has to go on until every bone, every muscle, every organ is got the right shape in the right place. And so in a binary hierarchy, there's twice as many cell fate decisions as there are cells in the final organism. So let's just say tens of trillions of cell fate decisions. That number is 10,000 times, 4 orders of magnitude higher than the digital information content of the human genome. So extraordinary that, so our DNA is largely devoted to controlling development. The way it does it is to produce RNAs that organize the genes to express both proteins, but also other regulatory RNAs in what the engineers call a feed-forward system that decides that where every cell, informed by environmental feedback, but where every cell has an internal program that says, what do I do next? Do I divide, or do I differentiate into a muscle cell or a bone cell or whatever? So the fundamental misunderstanding was to assume that all genetic information is transacted by proteins, largely true in microorganisms, not true in us. Of course, the proteins are critically important. If you have a mutation in it, you're in big trouble because they're the Lego parts. In fact, that's a good analogy. If you imagine you were given a set of Lego parts for Christmas and a set of plans, and the plans are to build a house. But another set of plans could be to build a castle, and there'll be a few more parts in a castle. You might have a moat and a few other things, but by and large, the components are common. And so the complexity of the thing you can build from a fairly core set of component parts is really determined by the plans, more so than the parts themselves. And in fact, that's the other thing I should have mentioned. The other shock that came along was to find when the genomes were sequenced that we have 20,000 protein coding genes, which was much less than people thought. But so do the nematode worms in the soil that are only 1 millimeter long that you can hardly see, only have 1,000 cells. And what's worse, nearly all of those proteins are orthologous, have similar functions. So the Lego part set for animal development goes back hundreds of millions of years. As complexity evolved, most of it was driven by the complexity of the differentiation development pathways, the cell fate decisions. But of course, along the way, new components were invented, say for neuronal transmission or whatever. So that's the narrative.
So part of the fundamental misunderstanding came from going from models of how bacteria DNA worked to animals and humans.
Yep.
And it was a mistake.
Yeah, so you could say, well, genes make proteins. That's a true statement, but that to extend as they did to think that all or most genetic information is transacted by proteins was the mistake. So you look back at the history of science, and I've written an article about this on Kuhnian revolutions. You remember Thomas Kuhn, the structure of scientific revolutions. Mostly what is understood or thought to be understood is not incorrect, but it's vastly incomplete. So to go from Newtonian to Einsteinian physics, or from Ptolemaic to Copernican, solar systems, well, that was a mistake. But mostly it's that there are things that people hadn't expected and didn't, And the tragedy of science here is that the surprising results are usually swept under the carpet because people are so wedded to their current understanding, not just in terms of the understanding, but in terms of the sorts of investigations they do. They're living within that framework, that paradigm. And they get quite defensive when you suggest that the framework might be wrong or incomplete. And that's happened over and over again in science. So I have to say science I find incredibly conservative. Now, that's good in some ways. But somebody once said, the best science is done at the point of greatest surprise. And that's true. In fact, I tell my students, look for the things you didn't expect. Now, if you get a result that you didn't expect, you could have just made a mistake, but you go back and repeat it. And if it repeats and you're careful, then you say, something's going on here that we didn't expect. And that's the window to discovery. As Leonard Cohen once said, the cracks are where the light shines in.
Absolutely.
Yeah.
Do you think some of the trying to think. The difficulty of accepting the idea, or even the gradual acceptance of the idea, came about through things like metaphors with software instructions and things. Gradually, people got the idea that it's not enough just to have the Lego bricks. You've got to have instructions on how to put them together.
Yeah, it's slowly. I don't think it was a big influence because,
Mm.
I mean, culturally, and this is a problem in my field, I think, its birth was more it was a mixture of biochemistry and genetics. We go back to, the first genes at Lackoper and things. But it's intrinsically reductionist. And most people are interested in what this protein does, what that pathway does, often in the context of disease, understandably. But they don't think about how the system works or is put together. And in my, I was going to say criticism, not criticism, observation is that people still rooted in biochemistry, structure, which is important, but they don't think about information and how the information is organized and transacted. And that's what you have to do when you come up to understanding how organisms are built, and so I think that was the biggest problem. You could give the analogies all day long and it would occur to people, appeal to some people, but most people are skeptical, they're in their own trench, By the way, the interesting sidebar here is firstly that the vociferous resistance to this, and because I watched over years, and occasionally you've got to respond to it, when it's given coverage in Nature or something, is commonly the ones who are most vociferous in their opposition to and defense of junk DNA are people who are quite aggressive anti-intelligent design. Now, if you think about that, they use the junk DNA argument to argue against intelligent design because an intelligent designer wouldn't have put all that junk in.
Right.
So the idea that it's not junk destroys that argument, and they don't like it. So
no.
I don't like ideologues on either side of this fence. I'm not pushing intelligent design because I can easily, in my mind, I've written about this, rationalize how evolution could have done this.
Yes.
But actually, it does provoke certain things. So the first is that I don't think evolution could work the way people think it does because random searches are not efficient enough to navigate the space. And I tried to do this in computers years ago, and the computer blew up. So I'm convinced, and there's evidence, that evolution has accidentally discovered ways to improve its search efficiency. Doesn't know, it's not prescient, doesn't know where it wants to go or what the possibilities are, but it can search the space more efficiently than just random mutations, So I think, and this also goes to Lamarckism, because Lamarck got thrown out by the theoretical biologists in the 1930s because they didn't want to pollute Darwin's ideas, although Darwin was quite happy because he didn't know what the mechanism of variation was. But there's now really a lot of evidence, and maybe I can talk about this in the evening, of intergenerational epigenetic inheritance. In other words, there is soft-wired inheritance that's dependent on previous experience, whether it's physiological or neuronal. We have evidence of both that actually conditions us as well. And that explains a lot of other mysteries, including missing genetic variation in complex disorders and so forth. So, I think we used to say the second half of the 20th century was the time of biology and computing. That was wrong. As far as biology is concerned, it was just a warm-up. This is the century of biology and medicine because we're just now starting to have the tools to get under the hood properly, and particularly into the brain, where a lot of this transgenerational inheritance is coming from too. So, and that has important, not only scientific, but also social consequences. In other words, if, I don't know if this is true or not, but let's just take a toy example. If you're abused as a child, then that may leave a scar on your children that you didn't expect, even if you've recovered and live a relatively normal existence. So there's all sorts of wonderful things in our genome. I'm convinced that it's the most sophisticated information suite that you could possibly imagine, and that we can, understanding the way it controls tens of trillions of self-fate decisions with perfect accuracy, because identical twins are phenocopies, will open our eyes to all sorts of possibilities in terms of how we program other systems and also artificial life.
there
But that's
was.
down the track.
Oh, sorry. No, that's really interesting because I come across years ago some of the examples of where there seem to be epigenetic changes occurring in a short period of time. Was it the
Mhm.
quagga? I think there was. No, I've got it wrong. There was some sort of African horse type.
Oh, you're talking about the fish,
Ah,
stickleback fish. Yeah,
okay.
yeah, yeah. So these and people tend to brush this under the carpet, whereas I think if you're a good scientist, you should be curious. Now, you always got to think of the flaws in your design or interpret. But I think there are lots of compelling examples now in mammals. For example, if you fear condition mice, like it's a well-established paradigm. You don't hurt them too much, but when you ring the bell or puff a perfume, you give them a cold shock. So they're standing on a metal platform, and all of a sudden it goes cold. It's not too unpleasant, but they get a bit of a fright. That's only got to happen once or twice, and next time they hear the bell, they jump, they freeze. Because they're expecting something unpleasant. So there have been experiments to show that 3 or 4 generations later, you ring a bell and the mice freeze.
Mm.
And this is sperm inheritance, so you don't have the inter-ovary issue of transgeneration and that's what's bedeviled the Dutch study. I don't know if you're familiar with the Dutch famine. There's been
Yes.
a multi-generational consequence. The trouble is that the granddaughters of the women who experienced the famine
Yes.
were in utero at the time of they experienced the famine because
Ah.
they had babies, girls, but the ovaries in those girls were developing with the girls in the mother.
Right.
So you got to wait for another generation. But if you do sperm in experimental paradigm in mice, then you don't have any of that complication.
Mm.
So that's not the only example, but there's quite a few. And the mechanism, there's hints of mechanism as well about that. So I think we've got a long way to go. And really, the lesson for me, Ian, is that you've got to shed your preconceptions.
Mm.
Now, we all have them, and they're very hard to shed. But I think to say when something doesn't fit, you don't push it aside or try to plug a square peg into a round hole. You say, oh, that's interesting.
Yes.
And now when you're younger and more receptive to this, and that's why often the best science is done with people when they're young, because they're more plastic in their understanding of the world. But the problem, and I point out to my students, postdocs, is that you're still learning. So often when you think something doesn't feel right, when you look into it, it's just you didn't understand enough, and you're satisfied that there's a cogent answer within the current understanding. But always keep in mind that the things that don't make sense is where the real opportunity is. Yeah.
And there's surely huge implications if you've got mice who've got something they've learned has gone into the generations. That's a giant paradigm shift. It's not just physical attributes if they know something.
Yeah. We don't know the limits to this. But my bias, if that's the right word, is that specific learning, like the history of the German Republic or something, that's unlikely to be inherited. Whereas, and I could talk more about it, but in more detailed levels, the microsatellites that actually expand and contract, which may be controlling gene expression patterns that then, but we don't know, and it's really a great new frontier. And of course, you've got instinctive behavior as well. So, if you put a toy orca into a goldfish tank, the goldfish are terrified.
Ah.
Yeah. The orca is an ocean mammal. Goldfish are freshwater carp, but they're imprinted. It's like, putting a snake in front of a cat or whatever.
Yeah.
So, and so nobody's ever pulled this apart genetically. And by the way, another one of my aphorisms is genetics discovers and biochemistry explains.
Ah.
Biochemistry discovers almost nothing. There are a few exceptions, but the clue has nearly always come from genetics, from the strange phenomenon that people went big into. The entire gene cloning revolution came from genetics, and identifying. And so the CRISPR revolution, in a sense, that was genomics and genetics. But so, sorry, I'm sort of digressing slightly.
That's all right.
But there's a lot to discover, and we need better tools. So every time we get new tools, we see more, whether it's imaging or, and that goes back to Leeuwenhoek, etc. But we also have to expand the way we think, and not assume that what our understanding is correct. By the way, and when Thomas Kuhn wrote about the structure of scientific revolutions in the early 60s, he said that there are two components to this. One is so we have a current understanding, and people work within that current conceptual framework or paradigm and do what he called normal science. Normal science. That is, that your experiments are designed and interpreted, or data is interpreted, your observations are interpreted within the framework of that paradigm. And that persists until two things happen. The first is that anomalies accumulate. He called them anomalies that don't fit. But you need a few of them before people start to take notice, because they, so the accumulation of anomalies. But then the second requirement before the paradigm changes is that there has to be a cogent alternative explanation that respects and incorporates the data of the past, but into a new framework that fits the anomalies as well. So you need a replacement paradigm before people will accept that something's wrong. They might suspect it, but the shift is only made when you've got a cogent explanation and a new framework. So,
yes, it makes sense in a way. I guess people want to have a framework ready rather than just go into, well, we just don't know, from when they thought they did.
Well, that's where they park it. Most of my colleagues out there studying protein X or Y, oh yeah, they're sort of mildly curious, but unless they've got some way of understanding it, then it's just, we don't know, and who knows whether they're right, the junk people or the other people. But when there's a framework that is cogent and makes sense and the evidence supports, oh, right, and now I get it. In fact, when I actually say to people here at the university that most genes are coming from genetic loci called enhancers, they've been known for a century and they were thought to be something else. And they're controlling cell fate decisions during development, and the mechanism is vaguely understood. People say, oh, right, now I understand. Okay, that makes sense. I get it, and everything's fine. Yeah.
Here you go. So people who want to innovate need to pay attention to the things that are funny and unusual and interesting that are often disregarded.
Yeah. In fact, I just did a podcast there's a really nice one called Night Science that you might have heard of, run out of New York and Dusseldorf or somewhere in Germany. And it's based on what creativity, And so that's the clue, is to look for the anomalies and don't ignore them, prosecute them. As I said, they may have an explanation, but until you find it. The second thing is that when I learned this, as I said in the podcast, from my mother, she was one of 5 sisters of Irish descent, and we had no money, they had no money. And so their entertainment was to debate things, argue. One said black, the other one said white, but it was always done with great wit, good humor, and I learned from these women, not only because, as I also tell my students, you don't learn anything by agreeing, you only learn by disagreeing. But you've got to do it nicely, and not be offended because somebody's got a different view, whether it's about politics or anything else. I mean, it's silly to take a moral posturing on a point of view. You've been asked to explain it, justify it, what's the evidence, what's the logic, So one of the things I learned early was this, someone said black. I said, well, hang on, maybe it's white. That's where the junk is. So somebody told me in a pub in Houston, Texas, that it was junk. And I thought, I just thought, maybe it's not. So I think being a contrarian, but not destructive one, but is very useful to look at it from another point of view. So if it doesn't make sense, question the assumptions behind it and look for an alternative explanation that might make sense.
Do you have an opinion on, way back, I think it was, there was a Professor Malcolm Simons who patented or tried to patent junk DNA?
Yeah.
Um,
No,
do you have an
Malcolm? Yeah.
opinion on that?
Yeah, look, in fact, Malcolm used to come to see me because he was scared that my work would interrupt his patent. So it didn't say the problem. So what Malcolm recognized was that for protein coding mutations, and that's at that time, the only technologies available to detect them were restriction enzyme cleavage sites. The sequence change would either create or destroy a restriction, for these sequence-specific enzymes. But finding those was almost impossible because the mutation in, say, the cystic fibrosis gene, didn't create or lose a site for any of the enzymes. So what he realized was that nearby in the non-coding, the regulatory sequences, both the introns inside the genes and the intergenic sequences were full of variations. And they were tightly linked so that if the version of the gene that was defective in cystic fibrosis couldn't be polled, but a tightly linked variant nearby in the non-coding, the junk DNA, could be identified by a restriction site, present or absent, then he had a surrogate marker that was tightly linked to the normal or damaged gene. And you could use that as a diagnostic. Very clever. Now, what happened in the patents of this was that this idea of genetic linkage was well established in genetics, going back a century to Drosophila, Morgan, all the early stuff on gene mapping. But the biochemists, who were the gene cloners or the people in the patent office, hadn't been trained in genetics. So they thought this was terribly innovative because you had a closely linked variant that could be analyzed by the technology at the time. So at one point it was clever, but it wasn't that inventive in the sense that the geneticists knew about this. I give Malcolm credit for putting the two together. Good on him. And they made some mileage out of it for a while.
Now, I'm aware you're going to have to go soon.
Yeah,
Would you
I've
like to
still
make
got
some
10 minutes or
closing?
so.
Oh, okay.
Sorry?
Is there something else you'd like to talk about before you have to go?
No, I think I've covered it. I mean, just to recap, Ian, I think the assumption that genes only make proteins turned out to be wrong. The patents will say, oh, some genes make ribosomal RNAs, but they're the infrastructure of making proteins. But the genes are synonymous with proteins turns out to be true, largely true of microorganisms, but not true in us, that most of our genes are actually producing what I call organizational RNAs that control our development. And that's obvious in retrospect. That's a good test, by the way. Retrospective obviousness is a good test of whether you're right or not. So the broader lesson is that one should always be aware of the assumptions that are underpinning things, and that if observations are made that don't fit easily, question the assumptions and look for alternative explanations. But I think it's also testimony to both the conservatism and to some extent boringness of science on the one hand, and the other, its ability to get there in the end and make exciting new, have an exciting new perspective, which informs us in all sorts of ways. So, and I just don't apply this to molecular biology. I think all of the scientific disciplines suffer from this lurching from paradigm to paradigm problem. There's a Proctoral story, which I think is made up, but that Lord Kelvin said that predicted the end of physics in the 1890s. And then along came Rutherford and Einstein and Schrodinger and threw all the cats up in the air.
Yes.
So, so always question, always be curious, challenge. But I think one of the problems I think in current culture is that we've forgotten that challenging ideas and permanence perspective, whether it's, as I said, political or anything else, is actually a very good thing to do. But you've got to do it respectfully, but cherish it, because when you disagree, you actually learn.
Very important. Well,
Yeah.
on that note, Professor
Yeah.
Mattick, thank you so much.
It's a pleasure, Ian. Thank you.
That's
Bye-bye.
awesome. Thank